Welcome to yet another episode of Crash Course in Biology. Today we are going to discuss human reproduction. So this is the second year chapter. We are going through the different questions now. Right. So here we have the first question. The attachment of blastula on the wall of uterus is called. The attachment of blastula on the wall of uterus. You know, there are reproductive events. What are the different reproductive events you have to study? First one is gametogenesis. Gametogenesis, then there is insemination, then there is fertilization. After fertilization, there is gestation, and after gestation, before gestation, there is implantation, and then gestation, and finally parturition. So these are the different steps in the case of human reproduction. So you have to study it in a correct sequence, and also you have to identify the definitions of these reproductive events. The attachment of blastula on the wall of uterus is called implantation. Parturition, you know, it is another term for delivery or giving birth to a young one. Then gestation. Gestation is the development of embryo within the uterus or within the womb of mother. And micturition. Micturition is voiding of urine. Okay. So here the correct answer will be option B. Next question. The male germ cells are also called. Male germ cells are also called spermatogonia. You know, there are two types of cells lining the seminiferous tubule. Spermatogonia or the male germ cells, and also Sertoli cells. So these are the two cells lining the seminiferous tubule when you take a cross section. Right. So answer here will be option A. What about oocytes? Oocytes are formed during the oogenesis or formation of ovum. There are two types of oocytes, primary oocytes and secondary oocytes. So when you follow this oogenesis, you should keep in mind the haploid cells and diploid cells. Primary oocytes are diploid, whereas secondary oocytes, polar bodies, ovum, they are all haploid in nature. Now oogonia, they are the cells from which oogenesis will begin. All the oogonia will be formed within a female embryonic development. During embryonic development itself, all the oogonia will be formed. Then spermatids, they undergo differentiation to form spermatozoa. So here the correct answer will be male germ cells are also called spermatogonia. Right. Next, male germ cells and Sertoli cells are present in, as I told you earlier, they are present in the seminiferous tubule. In the lining of the seminiferous tubule, you can see these two types of cells, male germ cells and Sertoli cells. Seminal vesicles, they form the accessory sex gland in male. Vas deferens is part of the accessory duct, and vas deferentia that also form the accessory duct in male. Sometimes they will ask you to identify the accessory duct, or you will have a set of options from which you have to pick which one of the following is not an accessory duct. So all the accessory ducts, their sequence, they are very important. Sequence is important because through these accessory ducts, the male gamete will find its journey. So this question is very important. So male germ cells and Sertoli cells are present in the lining of seminiferous tubule. Right. Now we move on to the next question. One of the following loops over the urinary bladder. Vas deferens will loop over the urinary bladder. You know, in the textbook it is written clearly that vas deferens, which is formed from epididymis. Epididymis is the region where sperm cells are stored, and final maturation of sperm cells will happen in epididymis. And after epididymis, you can find this vas deferens. In this diagram, it is very clear. You can see here in this picture, the vas deferens will ascend to the abdomen and it loops over the urinary bladder. Right. and after looping of the urinary bladder, vas deferens will join with a duct from seminal vesicle. Then it forms the ejaculatory duct. So vas deferens, it is paired, and usually we are cutting the vas deferens in sterilization procedure, that you know. So here our answer will be vas deferens. So option D. Rete testis, it is within the testis. Within testis, arising from the seminiferous tubules, there will be rete testis. Tubuli recti, rete testis, like that. So first of all, there will be tubuli recti, then rete testis, then vas efferentia, and then comes the epididymis portion. So that is a sequence. So there is an intratesticular duct system or duct system within the testis, and also there is an extra testicular duct system or a duct system outside the testis. So here rete testis is within the testis. Vas efferentia is also within the testis. Okay, 10 to 12 vas efferentia will be the arising from the rete testis. Epididymis, I told you, for final maturation and storage of sperm cells. So this diagram is very important, right? So we move on to this next question. A duct from the seminal vesicle is received by. So in that diagram again, you can see vas deferens will receive a duct from seminal vesicle, and then only it will form the ejaculatory duct. So vas deferens will receive a duct from seminal vesicle. And remember, if there is an option like ejaculatory duct, what is ejaculatory duct? After receiving a duct from seminal vesicle, this vas deferens is now called, it is termed as ejaculatory duct, and ejaculatory duct will join with the urethra. So here answer will be option D. Option D will be our answer here. Again, the ducts are given in options. It's very clear. Now, which of the following is not part of female external genitalia? The part of reproductive organs which are visible outside the abdomen, they are called external genitalia. They have a role in reproduction in female. And clitoris, mons pubis, labia majora, labia minora, hymen, these are the external genitalia in the case of female. So here, one sentence each you have to by heart for these external genitalia. Okay. For clitoris, you can also say that it is homologous to the penis, which is found in male. Mons pubis is a hairy portion which is covered, which is having a cushion of fatty tissue. Labia majora, labia minora, they are fatty folds of tissues again. Cervix is the odd one here. It is not coming in the list of external genitalia. Cervix, it is a part which is found within the body of female. It is not visible outside. So cervix or cervical canal, it is the odd one here. It is not part of external genitalia. So answer will be option D here. Now we move on to next question. Which of the following is not a male accessory reproductive gland? There are 3 accessory sex glands in male. What are they? A pair of seminal vesicles, a pair of bulbourethral glands, and a single prostate gland. Remember, prostate gland is single. Here, seminiferous tubules, they are not a type of gland, they are actually tubules. They have nothing to do with these accessory sex glands. So our answer here will be, which is not a reproductive gland. It is seminal tubules, seminiferous tubules. Seminiferous tubules, they are not forming an accessory sex gland. So answer option A here, option A. Coming to the next question, identify the hormones A and B. So you know, menstrual cycle is very much influenced by the pituitary hormones and ovarian hormones. The pituitary hormones are FSH and LH. They influence the menstrual cycle. Okay. What about ovarian hormones? Ovarian hormones are estrogen and progesterone. So here it is written as ovarian hormones, and in the picture, the graphical representation, it is very clear that The line A here, marked as line A, it is estrogen. Estrogen, it is produced from the Graafian follicle, whereas progesterone, it is produced from corpus luteum. So, in the proliferative phase, estrogen is high, and the secretory phase, progesterone is high. You got the point? So, answer here will be estrogen and progesterone. Okay, labeled as A and B, and the structure which produces B or progesterone will be corpus luteum. It is a yellowish structure, and after rupturing of the Graafian follicle, we call it as the corpus luteum. So the correct answer for this question will be option A. So sometimes they will give another picture, that of pituitary hormones, and they will ask the same pattern of question. So now we have the next question. Match the following. You have two columns, column A and column B. So here this question is about the different developmental stages in human embryology. As you know, human development or gestation can be divided into 3 trimesters. First trimester, it includes first month, second month and third month. Like that, the second trimester will include 4th, 5th, 6th, and the third trimester includes 7, 8 and 9. So like that, 3 trimesters you can see in human development or in gestation. Here, the different developmental stages are shown in this picture. So you have to identify the changes happening in each period. Here, in this question, you can match embryonic heart. Development of embryonic heart will happen in the first month itself. Okay, so 1 you can match with a S. Then limbs and digits. Limbs and digits are formed in the second month. Then external genitalia. External genitalia will be formed in the first trimester. You can identify the genital organs in the first month itself, first trimester itself. Then first fetal movements are observed in the 5th month. And like that, fine hair, eyelids and eyelashes are separating during the second trimester, or by the end of the second trimester you can see that development. So correct answer will be option C for this question. We move on to the next question. Choose the option showing the correct labeling for the following diagram in the sequence A, B, C and D. So here we have a set of options, and from the options you can pick option A. So this is an NCERT diagram. Remember, all NCERT diagrams are important. You have to identify, some questions will be asked to identify the correct labeling, and further they will ask the functions also. So here the answer is option A. Umbilical cord is marked as A. Then the placental villi you can see as B, and yolk sac is marked as C, and cervical plaque marked as D. So correct answer is option A for that question. The correct sequence in embryonic development. You know, embryonic development, it is having a sequence. After fertilization, a zygote is formed. As in this diagram you can see it very clearly. Then zygote will undergo repeated mitotic divisions. You can call it as the cleavage. And the cleavage will produce a morula initially. The 8 to 16 cell stage is called morula. And this morula further develops into a blastula. So that is the correct sequence. Answer is option A here. So this diagram you have to identify the stages also, right? Now, polyspermy. How you can prevent polyspermy? What is polyspermy? The entry of more than one sperm into ovum is known as polyspermy. So this is prevented by formation of a fertilization membrane. After entry of the first sperm cell itself, a thickening of the outer layer will happen. So the egg membrane will get thickened to form a fertilization membrane. A lot of chemical reactions are happening there. So as a result of that, there is formation of a fertilization membrane there. And this is altogether, it is coming under the reproductive events. So answer option B. So no more sperm cells can enter into the ovum once a fertilization membrane is formed. So answer option B. What about option A? Acrosomal enzymes within the acrosome, which is formed from the Golgi complex, which is part of the sperm cell. Acrosome is having sperm lysins. Sperm lysins are hyaluronidase, sonalizin, acrosin, etc. So these sperm lysins are helping to penetrate through the egg membrane. So sperm lysins, the list and also their functions you have to by heart. Now option C, enzymes produced by glands in oviductal wall. Capacitation. Capacitation is the applicable for the sperm cell. The sperm cell, it is getting activated, and that activation to fertilize the ovum, that step is called capacitation. So it is applicable for the sperm cell. So here answer option B. Okay, now coming to question number, match the following now. We have a set of options. You can match it very clearly. First menstruation is called menarche. So reproductive years of a female is from menarche to menopause. The first menstruation is called menarche. Then prevents polyspermy. As we discussed, it is by fertilization membrane. So one you can match with menarche 1S. Then prevents polyspermy, which is preventing polyspermy, fertilization membrane 2P. Then 3Q. Why? Because fertilizing is produced by the unfertilized egg. Okay, to attract the sperm cell over it. Then fusion of the pronuclei is called amphimixis. The two haploid nuclei after the sperm entry, they will fuse, and the fusion step is called amphimixis. Then first fecal matter is meconium. So 5 you can match with meconium or R. So the correct answer will be option B. Okay. Now we move on to the next question. Identify the timing of release of the second polar body. The second polar body, as in this diagram, you can see it is produced or it is released after the sperm entry and just before the amphimixis. So you know, during ovulation, it is not ovum released from the Graafian follicle. The ruptured Graafian follicle will release the secondary oocyte, and to this secondary oocyte actually the sperm enters. So that point is very important. Sperm enters into the secondary oocyte. So by that time already the first meiosis is over. Okay. So now waiting for the second meiosis. After the sperm entry only, there will be release of the second polar body, and after release of the second polar body, there will be fusion of the pronuclei or amphimixis. So second polar bodies are extruded or they are released after the sperm entry and just before the amphimixis or just before the fusion of the two pronuclei. So this diagram is very important. This is a diagram of oogenesis. Here you have to identify the different stages. They will ask you the function of polar bodies. Polar bodies are formed in order to conserve the cytoplasm for the ovum. There is cytoplasmic inheritance also. And you will have to identify the different haploid, diploid stages here. Okay. Which cell undergoes meiosis here? Primary oocyte. So after first meiosis, which cell is formed? Second polar body. After second meiosis, which cell is formed? Ovum. Okay. So not only ovum, there will be also release of second polar body. So always the division is unequal here. First meiosis and second meiosis, the division seems to be unequal. Now we have the question of hormones produced by the placenta. Okay, placenta is producing a set of hormones, and HCG, HPL and relaxin. These three hormones, they are produced only during pregnancy. So that is very important. So when you study the endocrine role of placenta, do remember that the placental hormones like HCG, HPL and relaxin from ovary, and they are produced only during pregnancy. So this question is very important, right? Now we have a diagram question. This is the diagram of blastocysts. So in the blastocyst, you can see an outer layer of trophectoderm, and you can see an inner cell mass. So labeled as X will be inner cell mass, and labeled as Y will be trophoblasts. As you know, the blastocyst will attach onto the uterine wall during implantation. So the trophectoderm part is actually binding with the uterine wall. So here function is also important. Inner cell mass will develop into the embryo, whereas the trophoblast will develop into the fetal membranes also. So here the correct answer will be option A here. Okay, now we move on to the next question. Which of the following is wrong regarding parturition? Parturition is the delivery process or giving birth to a young one. That process is called parturition. That is culmination of the reproductive events. So among options you can read, signals for parturition originate from the fully developed fetus and placenta. That is true. Oxytocin released from fetal pituitary. That is a wrong statement, because oxytocin released from the maternal pituitary, as a result of that, fetal ejection reflex will happen, because it will induce mild uterine contractions. So that is the beginning of parturition. So that is the beginning of delivery. So first of all, whenever there is a signal sent from a fully matured fetus and placenta, first of all, the maternal pituitary has to release oxytocin, and that will act on the uterine wall to produce mild contractions or fetal ejection reflex. So here answer is B, because that statement is wrong. Not from fetal pituitary, it is from maternal pituitary oxytocin is released. Then C option, parturition is induced by a complex neuroendocrine mechanism. True, because it is a reflex action, kind of or involuntary kind of a process. Then oxytocin acts on the uterine muscle and causes stronger uterine contractions. That is also true, because it is a positive feedback. More and more oxytocin, more and more uterine contractions will be the result. Then option E, mild uterine contractions are there for fetal ejection reflex. That is also true. So correct answer is option B, because that is a wrong statement here. Now we move on to the next question. In cleavage up to morula stage, you know, zona pellucida, which is the outer layer of the egg. The outermost is corona radiata, having follicular cells. But the zona pellucida will retain even after the fertilization. Up to morula stage, zona pellucida will retain. That is why up to morula stage there is no comparative increase in the size of the embryo. The size of the embryo will not increase up to morula stage. It is because the zona pellucida is retained in it. Okay. So blastomeres, they are getting smaller and smaller when it develops up to morula stage. Further increase in the size of blastomeres will happen after the breaking of the zona pellucida or after the morula stage only. So that is very important point. So here in the cleavage up to morula stage, size of blastomere decreases. That is a correct statement. Third statement is also true. The nuclear cytoplasmic ratio increases. Okay, because cytoplasm is comparatively low compared with our DNA. DNA will be multiplied. DNA synthesis will occur. Okay. So here, option second statement is wrong. DNA synthesis occurs during, of course, it occurs during embryonic development. Then fourth, size of the embryo increases as the cleavage proceeds from zygote to morula. No, size of the embryo decreases. So the second and fourth statements are false, and the first and third are true here. So the correct answer here will be option A. 1 and 3 are correct, 2 and 4 are wrong. So with this question we conclude today's session of crash biology, human reproduction. We have discussed a set of questions, and we will be back with yet another chapter tomorrow, same time. See you then. It's goodbye from Stephen Classes.